Hello, everyone, and welcome to our MaxCyte sponsored event titled A cGMP Compatible Non-Viral CAR T Cell Manufacturing Process. My name is Leslie Eschinger, and I am a Director of Marketing at MaxCyte. I will be your moderator for today's engaging session. Our speaker for today's session will be Andrew Mancini, Field Application Scientist at MaxCyte specializing in cell and gene therapy. Andrew supported Dr. Marston, Dr. Ian Steen, and Dr. Shai at UCSF with technical assistance and troubleshooting techniques to optimize their cGMP workflow. So he is very familiar with the completed work and novel approach at this laboratory. I would like to remind everyone that this session is meant to be interactive, so we encourage you to participate by submitting questions at any time during the presentation. Let's get started.
Hi, everybody. Thanks for joining. My name is Andrew Mancini. I'm a field application scientist at MaxCyte. Today, I'll be talking to you about work done at the University of California, San Francisco, focused on creating a cGMP-compatible non-viral CAR T-cell manufacturing process using the MaxCyte platform. I first want to begin with giving you a brief introduction to CAR T-cell engineering. I know that most of you present don't need any introduction to this field. However, to make sure everybody's on the same page, I'll just briefly go through what CAR T-cells are, how they are made, and where CAR T-cell manufacturing is going in the future. I'll then pivot that right into the work that I want to discuss with you today, which is how the groups of Jonathan Essenson, Alex Marsden, and Brian Shai at UCSF developed a hybrid SsDNA template that enables highly efficient non-viral CAR T-cell engineering. I'll first focus on a lot of the developmental data which went into creating these templates and validating that they're effective for CAR T-cell engineering. And then I will lead that directly into the relevant work in which they use this process to put together a full non-viral cGMP-compatible clinical manufacturing workflow using MaxCyte's GTX instrument in conjunction with these hybrid SsDNA templates. We'll leave time for about 15 or 20 minutes of questions at the end of the session. But of course, at any time, feel free to put your questions in the chat. Be happy to answer them as they come through. So I wanted to begin with talking a little bit about what CAR T cells are and how they are made. So I'm sure that most of you here know this, but of course, CAR T-cell therapy is based around taking a patient's own immune cells and then engineering them in a way so that they can be used as an anti-cancer therapeutic. The basic way that this works is that you will have a patient with cancer and you will collect the patient's T cells via a process called leukapheresis. Once you've isolated their T cells, you will then go to a series of steps to manufacture the CAR T-cell product. And so CAR stands for chimeric antigen receptor. And what this does is that we're taking the patient T-cells, we are enriching them and activating them so that they begin expanding. And then we are putting in this chimeric antigen receptor into the cells so that it is stably expressed by the cells. And this CAR construct enables the recognition of a specific tumor antigen on the surface of the cancer cells, which will then allow the T cell to specifically target and ideally begin killing the cancer. Now, once you have stably introduced this CAR construct into the patient T cells, historically done using lentivirus, you can then expand these CAR T cells to hundreds of millions and billions of cells, and then isolate the final product, purify it, and dose it back into the patient. And again, these T cells have now a receptor, which is specific to something on the tumor. So something like CD19 or BCMA, which is typically targeted for hematopoietic tumors. And now these T cells can be highly effective in treating and even curing the cancer that the patient has. Now, as I said, historically, CAR T cells have been made using lentiviral integration of the CAR construct. So lentivirus has been used to introduce the CAR construct into T cells, because lentivirus will integrate into the T cells and you will have stable expression of this CAR molecule, no matter how many times the T cell expanded, both during the ex vivo manufacturing process, but also once it's dosed into patients. Of course, that means that you will always have a highly effective drug product, and you don't need to worry that much about the CAR receptor no longer being expressed on your T cells. So while that all sounds great, there are a lot of challenges involved with using viral vectors to manufacture CAR T cells. And a lot of that is due to the complexity and costliness of manufacturing viral vectors at a scale and under the regulations needed so that they can be used to create CAR T cells that are going into patients. So on this slide, I put up kind of a simplification of the entire process, not just the CAR T cell bioprocessing, which you saw on the previous slide, but also the viral vector bioprocessing, which needs to happen to generate the CAR vectors, which are used for stable integration. You can see this is a quite complicated process, and there's a very complex supply chain and overall amount of work that goes into this dramatically increases the overall cost and time for manufacturing CAR T cells. And so in the previous years, there's been a significant focus at looking for alternative methods for manufacturing CAR T cells that either don't rely as much on the viral vectors or completely gets rid of the viral components. And we call these methods the non-viral CAR T cell engineering. That's what I want to focus on for this talk today. And so what if you could just completely remove this entire block in red here? And instead of going through this entire viral vector bioprocessing, you can replace it with a construct which could be introduced into cells via transfection instead of through virus, and overall simplify your CAR T cell manufacturing. And so many groups have begun to go this route and develop a variety of CAR T cell engineering approaches that are either partially or fully non-viral. These involve transposase-mediated CAR integration using transposases such as Piggyback or Sleeping Beauty in order to have a stable, semi-random integration of the CAR construct into T cells. There's also non-integrating methods such as persistent CAR DNA vectors, such as epivectors or SMAR vectors, which do not actually integrate into the genome, but will stick around as episomes and allow for stable CAR expression, even through many cell divisions. You also have more transient approaches, such as CAR mRNA expression, where you take the CAR mRNA, you transfect it into your T cells ex vivo, and then in the time frame through which the CAR is expressing, sometimes 72 to 96 hours, you then dose the patient with those CAR cells. Now, where a lot of focus has been more recently is developing nuclease-mediated CAR knock-in methods. And when I say nuclease-mediated, I'm talking about using something like a zinc finger nuclease, a TALEN, or more commonly CRISPR-Cas9 in order to introduce a donor template containing your CAR in order to specifically integrate your CAR construct in a site-directed manner. And sometimes you are delivering this donor carrying your CAR with AAV. That is what is more of a hybrid non-viral and viral approach. But more and more often, you have groups using synthetic DNA templates, either small oligonucleotides or longer single-stranded or double-stranded DNA, in order to introduce the CAR construct stably without relying on virus at all. And when we compare any of these approaches to the viral approaches, what you see is that overall, for transposase-mediated integration, even the non-integrating approaches, but especially for the nuclease-mediated CAR knock-ins, there's plenty of advantages that these approaches have compared to the viral approaches, such as high expression, stable integration, the reduced immunogenicity, and a lower risk of random integration compared to virus, as well as simplifying the manufacturing process, removing the whole viral supply chain issues, and typically reduced costs. However, compared to virus, typically these non-viral methods will have historically lower transfection efficiency and lower CAR positivity compared to the viral methods. So while you might end up with 60 to 80% CAR positivity using a lentiviral approach, many times non-viral approaches will struggle to get over 60% CAR positivity in your final product. And so while this is specific to transposons, you can really apply both these benefits and negatives to any of the non-viral methods, which has groups searching for ways to improve non-viral CAR T engineering in a way that's able to match the efficiency of viral approaches. And one way groups are doing this is leveraging novel CRISPR-mediated knock-in techniques for non-viral CAR T engineering. And that's what the work I'll be talking about today focuses on. Now, before jumping into that, I just want to give everybody a quick refresher as to what a CRISPR-mediated knock-in is. So sure, most of you know, when you deliver CRISPR machinery to a cell, you're usually taking either the Cas9 mRNA or protein, and Cas9 is a nuclease, and complexing it with a guide RNA, which is directing it to a specific genomic locus. You put these into your cell using a transfection method, typically electroporation, either using the MaxCyte electroporation platform or one of the various other platforms that are available. And once the Cas9 nuclease and the guide RNA is in the cell, you will have the Cas9 enzyme introduce a double-stranded break in the genomic DNA at the site specified by the guide RNA. Once this double-stranded break is introduced in your target of interest, such as the TRAC locus, so it's the T cell receptor locus, you will have the repair of the double-stranded break occur through one of two methods. This could be either non-homologous end joining, NHEJ, shown on the left in this figure, where you have a slightly random repair of the double-stranded break, leading to small insertions or deletions that occur during the NHEJ process. This is typically used for gene disruption, say if you want to knock out a specific locus. But if you want to knock in a piece of exogenous DNA into the genome, you instead try to focus on the homology-directed repair pathway, or HDR, where you are supplying a donor template that has some amount of homology to your target of interest, as well as a piece of DNA that you're looking to integrate at your cut site. This can be a small piece of DNA, only 5 to 10 base pairs, or it can be a larger construct that's multi-kilobases, such as a CAR construct. When you deliver the CRISPR machinery alongside this donor template to your cells of interest, you will have the cells begin to undergo homologous recombination, which will actually integrate your your donor DNA directly into the genome in a stable fashion. Now, when we look at how this is happening, you can see we have the Cas9 complex binding the target strand specified by the guide RNA that you used. And we have our HDR template with 2 regions of homology and our desired insert. When these are paired together, we have the homology donor template being used to repair that double-stranded break, thereby giving us our insertion. And so what we'll be focusing on today is methods as to which you can make this HDR process more efficient. Because in general, HDR, even when you stack the deck and use very good templates and very good CRISPR machinery, you will typically only have 8 to 10% of cells in your population integrating your target in their genome, especially when it's a longer stretch of DNA, like a CAR construct. Now, many groups have been working on ways to increase this efficiency, as I mentioned, in order to get it closer to viral integration. And one of these groups is the group of Alex Marzin at University of California, San Francisco. In 2020, the Marzin lab released a paper showing the use of modified repair templates that allowed increased HDR efficiency for the knock-in of CAR constructs in primary T cells. What they did is that they used a standard Cas9 guide RNA system, but delivered these modified DNAs, which were double-stranded linear DNAs, which had a DNA insert surrounded by homology arms, similar to what you would see for any standard HDR donor template. But instead of just leaving it there, on either end of the construct, you had truncated Cas9 target sequences, or TCTS sequences, which enabled the binding of Cas9 to the donor template, but in a way where the Cas9 was not actually cutting the template. And so what this allows for is that when you have the Cas9 complex to the DNA in the cell, and the Cas9 has a nuclear localization signal, or NLS, you will then get highly efficient uptake of your DNA template into the nucleus of the cell where the HDR is going to occur. And so now you're stacking the deck further towards HDR by not only having the template in the cell, but making sure it's getting to the nucleus alongside Cas9. And what this group showed is that this was a very efficient strategy to increase HDR efficiency and CRISPR knock-in efficiency overall for large constructs in cells like primary T cells. So instead of having 8 to 10% CAR knock-in, you could use this strategy to go above 20, 30, even 40% knock-in, getting a lot closer to what you're able to achieve using viral integration strategies. However, there's one thing about T cells that you should know. It's that they don't really like to have double-stranded DNA in them. And while this was an effective method to increase the CAR knock-in efficiency, it had the downside of being quite toxic to the T cells they were trying to edit. And at especially higher concentrations of the TCTS donor template, you saw quite a bit of toxicity due to the presence of this exogenous DNA in the T cells. And overall, this limits the efficacy of this approach because you are essentially at a hard cap for the amount of DNA that you can deliver for the homology-directed repair. Else you have too much toxicity in your population, and you are killing off any cell which might even be undergoing the CRISPR-mediated knock-in. So, of course, the work and innovation didn't stop with these double-stranded templates. The Marsden group, alongside with Jonathan Essensten's group and Brian Shai, went back and tried to find ways to continue to optimize this approach in order to enable even more highly efficient non-viral CAR T-cell engineering. And this is where these new hybrid SsDNA templates come into play. And so the work that I'll be showing you in the coming slides is from this preprint, which is available on bioRxiv, titled Hybrid Single-Stranded DNA Repair Templates Enable High-Yield Genome Engineering in Primary Cells for Disease Modeling and Cell Therapy Manufacturing. Again, this is from the groups of Alexander Marsden, Brian Shai, and Jonathan Essensten, along with all of the collaborators and co-authors also shown up on this slide. And the overall goal was to find ways to improve upon that initial double-stranded DNA sequence with truncated Cas9 targeting sites, and in order to allow for even higher efficiency genome engineering without the toxicity in primary immune cells. And so what the group did is design a second generation of these repair templates using single-stranded DNA instead of double-stranded DNA for the homology-directed repair. And the reason why single-stranded DNA was used here is, in general, while T cells and other immune cells don't like double-stranded DNA, they will tolerate single-stranded DNA up to quite high concentrations, meaning you could potentially load in more of your template using single-stranded DNA compared to using a double-stranded DNA donor, and consequently, potentially could have higher rates of CAR knock-in using the strategy. And so what this group did is they took the double-stranded templates with the CTS sites on either end and adapted them to a single-stranded model where the center, including the insert and both homology arms, was single-stranded DNA. And then because Cas9 needs to bind double-stranded DNA, they have double-stranded regions on either side, which has the truncated Cas9 targeting sequences. And so on the left here shows the variety of strategies which they tested out in order to see which would be most efficient. So these are labeled A through J. This corresponds to the x-axis on the plot on the right, with different amounts of the donor being given into the cells via electroporation. So 160 nanomolar, 800 nanomolar, or 4 micromolar from left to right. And on the y-axis, the knock-in efficiency at their locus of choice. And if we look at these different plots, you can see pretty readily that on the right-hand side, so H, I, and J, ones where you have the single-stranded center with the double-stranded edges, were the most efficient across a variety of concentrations. And with this approach, they were able to consistently get up to 30, even close to 40% knock-in at the higher concentrations. And we see, though, at the lower concentrations that this approach is quite effective, especially when compared to the single-stranded control. So the addition of the Cas9 targeting sequences on either side of the SsODN significantly increased the efficiency of knock-in, which they were seeing in their primary T cells. And overall, after trying out all these different approaches, they settled on using this construct J, which is, I think, what we consider probably the most simplified of all the approaches, where we have the single-stranded core containing the insert with the two homology arms, and then the CTS sites are made double-stranded just by the annealing of a 5' and 3' oligo. And so they then While this was initially used for small knock-ins, as shown in this figure, they then want to apply this for the knock-in of large constructs and directly compare it to the double-stranded approach, which was publicized in that 2020 manuscript, which I gave you a glimpse into on the earlier slides. And so this first strategy was to do a 1500 base pair knock-in of TNGFR into the IL-2RA locus. And this was done using the SsCTS templates, as they are called. With this knock-in strategy, they were able to achieve up to 78% knock-in of TNGFR. And importantly, not only was the knock-in efficiency higher compared to the double-stranded approach. So at the double-stranded approach, you're capping out around 40 to 45%, whereas with the single-stranded approach, you're up to over 70%. We can see that at these higher concentrations of the HDR template, we are maintaining significantly more live cells. So we are able to avoid the toxicity of the double-stranded DNA CTS template, while even improving further on the knock-in efficiency. And overall, you can see on the right-hand graph here that we are able to achieve significantly more CAR T cells with the knock-in across the entire range of concentrations compared to either normal single-stranded DNA in the light gray, normal double-stranded DNA in the dark gray, or the CTS double-stranded DNA in the blue. This was not exclusive to TNGFR knock-ins, the IL-2RA locus. They also were able to efficiently knock in several other constructs, including the BCMA CAR shown here. This is an even larger insertion, 2,920 base pairs, and this is going into the TRAC locus. So this is now actually putting in a CAR construct into a relevant locus. And with this strategy, they're able to achieve 39% knock-in of the BCMA CAR with, again, higher efficiency compared to the double-stranded CTS templates and the regular single-stranded and double-stranded DNA. And we're able to achieve, again, significantly higher live cells and CAR T cells at the end of the process. So immediately it looks like the use of these single-stranded CTS templates pays off and can immediately circumvent the main issue with the double-stranded CTS templates, and that is the toxicity. And so, again, this being able to get around this toxicity means that you can use higher amounts of the HDR template for your process, thereby achieving higher knock-in efficiencies. And even more importantly, you can be sure that your cells are healthier throughout the process. So if you are making a cell therapy, you will have more active cells and more happy active cells at the end of the process, thereby likely leading to a more efficient therapy. So this was, I think quite exciting data to see, because now you have a really robust strategy that can be used for CRISPR-mediated knock-in in immune cells. And not only was this able to be done at one or two loci, they showed that across 22 different genomic loci, you could achieve comparable, if not greater, knock-in efficiency compared to alternative templates. And this demonstrates that, first of all, that different loci have different rates of knock-in, as I'm sure we all know based on genomic context, but that across the board, the approach using the SsCTS templates seems to be superior, for about 19 of these 22 loci, and for the remaining 3, it's comparable to the double-stranded approach. So again, showing the power of these SsCTS templates. And not only is this limited, and this is not only limited to bulk T cells, but you can use this in a variety of cell types in order to achieve robust knock-in, including individual CD4s and CD8s that have been separated out. Tregs, which are quite a sensitive cell type, responded particularly well to the CTS templates, as did HSCs, B cells, NK cells, and gamma delta T cells. All of these cell types tend to react negatively when double-stranded DNA is put into their cytoplasm. And so switching to the single-stranded DNA repair templates allowed you to achieve significantly higher knock-in efficiency across the variety of, all of the cell types shown here. And you can imagine that it's not just immune cells which don't like having DNA put into them. For any cell type which is sensitive to exogenous DNA, I would imagine that the single-stranded CTS approach would allow you to achieve higher efficiencies compared to any alternative approach using double-stranded DNA or unmodified single-stranded DNA. And so there's one additional, I would say, very cool thing which came out of this work, and that was going beyond just modifying the templates and actually looking at other strategies to which we could boost the HDR efficiency for the knock-in of large constructs into primary T cells. And this approach was looking at small molecule inhibitors, such as NHEJ inhibitors or HDAC inhibitors, which could be combined with the CTS templates in order to further boost the knock-in efficiency. And there's 4 drugs shown here: TSA on the left, NU7441 in the middle left, M3814 in the middle right, and XL413 on the right here, each with different mechanisms of action. But all 4 of these drugs at an ideal concentration shown in red were able to significantly boost the knock-in efficiency of the CTS templates using CRISPR in primary T cells, while having minimal, if any, effect on the cell viability. So now you can imagine this is another tool in our toolbox that we can use to begin making non-viral CRISPR-mediated CAR knock-in even more efficient. And so what these groups did was then take these drugs either by themselves or in combination and apply them to either the double-stranded DNA CTS templates or the new single-stranded DNA CTS templates, and ensure that these combos of small molecule inhibitors did indeed increase the knock-in efficiency for the template types. And as you can see here, for both the CD5HA knock-in and the CLTA-M cherry knock-in, the single-stranded CTS alone without drugs already has a significant increase in knock-in efficiency over the double-stranded CTS without drugs. But then as we begin adding in the small molecule inhibitors, either this MT combination of TSA plus M3814 or the MTX combination with all 3 drugs, we can improve this knock-in efficiency even further, both with the single-stranded and double-stranded templates. So again, this is another weapon in the arsenal in order to really increase the knock-in efficiency and I'd say overall flexibility of this non-viral approach. So everything I've shown you so far has been done at the small scale, using a small scale research use electroporation platform in order to really develop these templates and evaluate the feasibility of this approach. However, University of California, San Francisco has a big focus on translational research and actually getting the research into a stage where it can be used to develop treatments for patients. And the labs of Alex Marzin, Brian Shai, and Jonathan Essenson in particular are always focused on treating patients using the tools that they develop. So the next step in this work was to take the single-stranded templates which were developed and which showed previously can be used for Knockin and integrate them into a GMP-compatible non-viral CAR T-cell manufacturing process. And this is where the MaxCyte platform really came into this work. Because when you need to scale up to transfect hundreds of millions or billions of cells, and you need to do it in a way which is gentle and also with an instrument which is cGMP compatible and has the backing of an FDA master file and is clinically validated, what many groups do is use our MaxCyte GTX electroporation platform for their non-viral engineering. And so what the Marzin, Shai, and Essenson labs did was use the MaxCyte GTX platform in order to deliver the CRISPR genome editing machinery alongside the SsCTS templates as part of a CAR T manufacturing process. And this was using the BCMA CAR, which I showed previously, in an autologous process, meaning that you have T cells isolated from the patient. These were then activated and expanded initially before the electroporation of a CRISPR RNP, so the Cas9 protein plus the guide RNA, alongside the SsCTS template for the BCMA CAR. These were then expanded for a further 8 days until they would then be dosed back into the patient for the treatment of multiple myeloma. Now, what they were able to achieve using this GMP compliant process is, in my opinion, quite remarkable, because everything which was done in the process which I just showed you was done in a way which it'd be able to be translated directly into a manufacturing space. Again, that's why we call it GMP compliant. And rather than seeing a drop-off in the efficiencies when we did the scale up to a clinical scale, so transfecting now 100 million cells instead of just a couple million cells, and doing this in a way where everything is GMP compliant, we were able to not only maintain the knock-in efficiency, which was done at the small scale, we were actually able to significantly improve upon the knock-in efficiency. So previously at the small scale, it's typically around 30 to 40% for the BCMA positivity through the non-viral CRISPR knock-in. Now, in the setting where we're just using the SsCTS template with no additional small molecule inhibitors, at the clinical scale, we were able to achieve up to 46% knock-in efficiency in primary T cells at day 10, end of the process. We were able to improve upon that further by adding in either M3814 by itself or the MT combination, so M3814 plus TSA. And this allows us to achieve over 60% knock-in efficiency at day 10. And this 60% barrier is what many, many groups have struggled to reach, even at the small scale, because this is really comparable to a lentiviral approach. And not only were we able to achieve over 60%, we did this in a process which could be used to actually manufacture CAR T cells for clinical use. And importantly, when we look at these cells throughout the process and at the end of the process, we see three key things. First, we're able to achieve the estimated patient dose that is needed. And so on the left here, we're looking at the CAR positive cell count. So these are cells that are stably expressing the BCMA CAR, either at day 7 or day 10 of the process. And in the black gray line at the top, this is just the standard Knockin with no inhibitors. And you can see we are able to achieve up to about 1.5 billion CAR T cells at end of process. Now, when we add in the small molecule inhibitors to increase Knockin efficiency, we do see a reduced expansion of the CAR positive cells. However, even with the double small molecule inhibitor combo, we are still exceeding the estimated patient dose at the end of process. So we have multiple strategies to achieve the amount of CAR T cells that we actually need to treat patients. And when we do immunophenotyping of these CAR T cells, we see that in all three conditions, these are enriched for the TSCM subpopulation. So that's the stem cell memory T cell phenotype. And that predicts highly effective T cell therapy in vivo. And to cap this off, when we do the in vitro killing assay, no matter whether this was done with no inhibitors, with a single small molecule inhibitor, or the double combo, all three strategies were able to robustly kill BCMA positive target cells. And so overall, from this process, not only were we able to achieve very good CAR Knockin, we were able to maintain good cell expansion, achieve favorable T cell phenotypes, and ensure that the cells are functional when aiming to kill their target tumor cells. And so I will end it there and leave you with a few conclusions. First, I hope that this showed you that the use of single-stranded DNA hybrid repair templates, also known as SsCTS templates, is able to significantly improve the efficiency of CRISPR-mediated CAR Knockin, not only in primary T cells, but also in any cell type which is sensitive to double-stranded DNA. Additionally, I hope that you took away from this that this SsCTS approach is quite flexible and can be used agnostically and in combination with small molecules for robust knock-in across cell types and genomic loci. So wherever and whenever you need to do a CRISPR knock-in, you can use the SsCTS templates with any of the small molecules shown here in order to really fine-tune your process. And of course, because we're always looking towards treating patients, importantly, using the MaxCyte GTX electroporation platform, we were able to scale up this non-viral CAR T cell process to a clinical scale in a GMP compliant manner in order to create a robust CAR T cell manufacturing workflow using the SsCTS approach. And of course, this is, in my opinion, the most important thing, because now not only have you done the proof of concept at small scale, but this is actively creating material which will go into patients. So this is a great proof of concept for combining a novel CRISPR knock-in approach and using the clinically validated MaxCyte GTX platform in order to create a fully non-viral CAR T cell manufacturing workflow. So I will end it there. And now I will stick around to answer any questions for the next 15 or 20 minutes that you might have. I hope you enjoyed and looking forward to hearing all of your questions. Thank you.
All right, great. Unfortunately, Andrew was unable to be live with us today to support with the Q&A. And in his absence, Jordan Dizon will a field applications scientist who works very closely with Andrew and is familiar with the body of work, will be joining us today to answer some of your questions. So I want to let you know if we can't address your technical questions during the session, we will ensure someone from MaxCyte or UCSF will follow up to assist with the answer. So I see quite a few questions have come in. I'm going to go ahead and get started by asking those. Jordan, are you on the line?
Yes, I'm here. Thanks, Leslie, for the introduction.
No problem. All right. The first question, what was the cell viability or live cell count for the control cells that didn't receive the single-stranded CTS?
Yeah, that's a great question. So if you guys take a second to look at the preprint, it's actually in the supplemental figure number 1. The live cell count for a non-HDR template control was about 25 million cells. And every single format, A through H, of those short hairpins or single-stranded combinations were all up between about 22 million to 30 million. So roughly, it was actually right in the middle range of all the different cell types. So there wasn't really a decrease in cell viability using the single-stranded templates versus the non-treated control.
Great. All right, I see another question here has come in. Does the single-stranded DNA also facilitate larger payload knock-in?
So they did try, the biggest payload they tried was the 2.9 kb payload, the BCMA CAR Knockin. If you wanted to try something larger, they do see that there's a decrease in efficiency quite rapidly once they go past 80 micromolar of the single-stranded CTS DNA template. So you could try to accommodate something larger, but you'll have to go down in the concentration ratio of the template. But the Marsden lab has not tried something larger than that 2.9 insert yet.
And can you explain the difference in the effect of the CTS addition and or the double-stranded DNA substitution with the single-stranded DNA for the 22 different genomic loci from no effect to a substantial improvement?
Yes, there's two critical components, right? If you're comparing the single-stranded DNA alone with the single-stranded CTS insert, that those Cas9 target sites really bind the Cas9 and then facilitate that entry into the nucleus. So that's really the main difference between the single-stranded alone and the single-stranded with the CTS template. Now, when we're talking about double-stranded DNA, right, the difference is mostly due to that reduced toxicity that you see when you have a single-stranded element opposed to a double-stranded DNA. So there's really two different things combining together that are really increasing the efficiency of that single-stranded CTS template.
Next question. Are the small molecule inhibitors approved for cell therapy?
Yes, they are. So everything that they used in this exact study has been currently in the clinic for other different types of drugs going through phase trials.
And how do these results compare to other groups using a lentiviral approach?
Yeah, so this specific study wasn't directly comparing a lentiviral approach. However, we have seen groups try both the MaxCyte electroporation platform in combination with a lentiviral transduction, so trying to compare the direct two in both CD8 positive T cells as well as CD4 positive T cells. And they overall see at least an increase in 15% of knock-in integration compared to the lentiviral transduction. So overall, we do see an increased efficiency using an electroporation approach.
Great. Next is, what is the ratio of the TCTS primers to the HDRT when preparing the mixture? Is it a 1-to-1? And is there an incubation period to prepare the single-stranded DNA HDRT mixed with the primers?
Yeah, so it is a 1-to-1 ratio. So you're correct. And then I think they did an incubation of 30 minutes at room temperature to really capture that.
All right, next question. Did the group do anything on off-target rates, and were there any safety concerns with the method?
Yeah, so I might have to defer to Andrew getting back to you on the specific off-target effects. There aren't too many safety concerns with this method. We do see overall that a non-viral approach is much better than a lentiviral approach when we talk about the end material coming out and how we're going to translate that to the patient. So overall, there's not too many safety concerns that we have to worry about.
Next question, are the chemical inhibitors that were presented continuously washed away at a certain time?
Do you mean were they presented continuously or washed away? Yeah, so they were actually they were actually introduced immediately post electroporation, and then they did have it continuing for about 24 to 48 hours post electroporation. So it was removed.
Great. And what is the cell viability after transfection, and how does electroporation affect cell viability? Is there any decreased cell viability percentage?
Yeah, so this exact, I kind of mentioned that a little bit before, but this exact, these constructs and this approach, they actually didn't see a decrease in cell viability too much post electroporation when we're considering the non-loading control. So basically you're electroporating, but you don't have that single-stranded template compared to all the other single-stranded templates. Those all teetered around 25 million cells post electroporation. All these samples were electroporated at 100 million cells per electroporation. So that was the starting cell count before we actually ran the electroporation event.
Great. Another question came in. Is the MaxCyte available for use in research as well as commercially?
Yes, that's a great question. So we do have it available for research. We have a lot of groups that purchase it for research up to their Ph.D. And then when they want to transition to the clinic, they go to more of a licensing agreement. But we definitely offer options for both academics and research scale industry partners to be able to use the system for purchase.
Great. And then what cuvettes or processing assemblies and or scale did the group use? And did they compare small scale to larger scale?
Yeah, that's a good question as well. So for MaxCyte, for the GMP component of this, they use the R1000 cuvette. So that's it goes up to a 1 mil reaction. They actually did electroporations for the volume of 600 microliters in that R1000 cuvette. And they electroporated, like I said, 100 million cells per electroporation event. And they did do smaller scale experiments. They used a different electroporation system other than the MaxCyte for the smaller scale work, actually. But it wasn't directly translatable and scalable to the GMP environment, so that's why they moved to the MaxCyte GTX system.
And you might not be able to answer this one, Jordan, but I will ask it. Is there a cost comparison that was done between the viral approach and the non-viral approach that you could speak of, or is that in the actual publication that Marcin and his group completed?
Yeah, so they do not do a direct comparison between the two methods in the system. I do know that we have a bunch of customers who have done that cost analysis before, and also members of our own sales team that have. So we could definitely follow up with you with more information on that.
Great. And is there any concerns with availability of and compatibility with GMP quality materials?
Yeah, so when we're thinking about GMP compatible material availability, it's been a little different right now with COVID, and we've seen that some companies out there are struggling to find materials. Without COVID in consideration, you should be able to get these GMP compatible reagents. However, right now we are seeing some longer lead times to get those materials. So if you are interested in doing a study like this, I suggest planning ahead of time, definitely, because we've got those COVID delays going on right now.
Great. And I'm seeing a couple more come in. This is another one about off-target rates, and we'll get back to you on that. Another one about safety concerns, and we already mentioned and addressed that. And then, well, maybe this will be the last one that we ask, although again, I don't know if you'll be able to answer this one, Jordan, but we'll try it out. Has the Marsa lab compared the immune phenotypes with the resultant CAR T cells and those products? And has the use of the double-stranded DNA knock-in templates varied compared to the viral vectors?
So they have done, directly, so you want to know the comparison between double-stranded DNA knock-in and viral vectors. They have not done that direct comparison.
Sorry, I meant with the phenotypes. Have you looked at the phenotypes of the resultant CAR T cells with the products from the double-stranded knock-in templates or viral vectors? And I think the answer is still no.
Yes, so they have compared the immune phenotypes of their single-stranded DNA knock-in with their double-stranded DNA knock-in, and those are very similar or the same, but they have not with viral vectors.
Perfect. Okay, I think that wraps up, and that's pretty much most of the questions. I think that we've had quite a few duplications, but I think that addresses everything. I want to remind our audience that the recorded version of today's talk will be made available soon after this live session, and you will be notified when it becomes available. And before I go, I want to take time to thank Andrew for all of his efforts in providing our viewers with this detailed presentation. To Jordan for filling in last minute with the great answers to the great questions that our audience has had today, to Lab Roots for hosting the events, and also to our audience participants for joining us today and for their intriguing question. This concludes our session for today, and until next time, have a great day.